Thanks for having me here. And time is running short, I guess, so I would rather be short and have a short look on the different vaccine types and some of the consequences, and perhaps also a little bit into the future. Has been said before, actually, that we somehow reinvent the wheel. So I will look at general vaccinology and try to then relate that to the development, research and development of vaccines against coronaviruses. And this is actually a list of the vaccines that are in clinical trials, have been authorized or abandoned. And it's without precedent that we ever had such a number of vaccines in parallel in clinical trials. More than 100 are now still in clinical trials. 19 had been authorized for emergency use or limited use in different countries. 9 have been approved for full use, and we know the major players very well on this field. And finally, actually, that's a surprisingly low number. Only 10 had been abandoned after the trials. Now, this is the list of the top runners. As we see, there are the RNA vaccines, obviously, which we are all well aware, Pfizer, BioNTech, Moderna. There are the ChAdOx and adenovirus vaccines, which take advantage as viral vector vaccines of adenoviruses. And currently, the only viral vector indeed is adeno, either ChAdOx or the adeno 526, where we don't have high neutralizing antibody titers before vaccination against adeno, obviously. Then we have the inactivated vaccines, mostly from China and also India. And finally, currently, there are 2 protein vaccines that have been approved, at least under limited conditions. The one from Novavax, which is now also available in Germany and being deployed in Germany, and the most recent addition from Baylor Institute in Houston, where I'm currently actually are together with the Indian colleagues, Biologic E. So there are a few more on the radar screen, or better say under the radar screen. There is a DNA vaccine already approved in India. There's a a polypeptide vaccine, a little bit similar to Hans Georg Ramsey's approach, but more simple, and that's been approved in Russia. And then there is a recombinant protein, soluble protein vaccine in Cuba approved. But for those, we have very, very little public information, so one cannot really say much about them. And surprisingly, there is no live attenuated vaccine yet in the field of coronaviruses. Most of us would think that the RNA vaccines are the most widely deployed vaccines, but in fact, actually, it's the canonical vaccines of killed vaccines, or better say, inactivated vaccines, that are actually globally deployed the most, followed by the adenovirus vaccines, and then finally the RNA vaccines, which clearly have so many advantages over the other one. Germany has a more simple picture. There are 2 RNA vaccines approved, 2 vector vaccines. Now, most recently, the Novavax vaccine has been approved, which is more or less virus-like particles, although they call them nanoparticles, with a very well-known adjuvant for immunologists, the ISCOM adjuvant, which they more or less use in a kind of modified version as Matrix-M. And the Valneva vaccine hopes for approval rather soon, within the next 2 months. Now, what you see here in the middle, and that's and that is a inactivated vaccine complemented with CpG for TLR9 adjuvant effects. Now, what you see here in the middle is actually the different types of vaccines in a very simplified form. And I would like to go very briefly what a vaccine really has to do. Obviously, it has to have antigenicity, that's very trivial. And then antigenicity is not always the same as immunogenicity. So you would want to have either inbuilt whole cell components or you do that with added vectors, but mostly it's of course adjuvants for the canonical vaccines. And then there may be some other components that may be neutral, beneficial, or harmful. So we all agree that the spike protein, which is more or less in all vaccines currently, is a central protective antigen, and therefore it is the major target of subunit vaccines, be they genetic like the adeno or the RNA vaccines. It contains the receptor binding domain, it contains the fusion sequence, which allows fusion of viral and cellular membranes. Unfortunately, it's highly labile, but that fortunately had been solved in the very, very beginning of research and development in the field of coronavirus vaccines by stabilizing mutations. But we need more, obviously, and I will focus here on the inactivated vaccines and the subunit vaccines, and these are the adjuvants. And the list of adjuvants is rather limited, actually, currently. First, of course, aluminium is more or less in all but the Nova vaccine, a central factor. It's good to stimulate antibodies. And as we know, it's not very helpful when we want to stimulate T cells. So this has been then complemented by TLR ligands 7-8 for the Bharat vaccine, inactivated vaccine from Bharat Biotech in India, and the recombinant protein vaccine developed by Baylor, by Peter Hotez, and then licensed without any financial interests to biologically in India. Then we have the kind of TLR9 vaccines here in the Baylor as well, and also Valneva uses a TLR9 agonist. And finally, the Matrix M and ISCOM-like molecule in Novavax, I think a very, very elegant adjuvant that they use, and the aluminium in Sinopharm and Sinovac. And I would guess that the alumin only in Sinopharm and Sinovac inactivated vaccines makes them rather poor in T-cell stimulation, and therefore also insufficient for the fight against coronavirus variants. And that's very disappointing because, as I said before, they are the most deployed vaccines currently. Now, the adjuvants, as I said, aluminium in any form is insufficient for the T cell response, gives you a nice Th2 cell response, and it's even not very strong in using, in stimulating non-neutralizing antibodies. The liposomes is an example for the ISCOMs, but also all of the adjuvants from the GlaxoSmithKline, the adjuvant series 1 here. And finally, we have the emulsions and Montanide, which Ansgerov is currently using, which is more or less a mineral oil, and therefore, as he said, resembles quite a lot the Freund's adjuvant type, is one of those that have been used as well. The AS03 is another emulsion which is actually used by Sanofi GSK for their recombinant vaccine against coronaviruses. To further stimulate, obviously, we have several vaccines that have added agonists for the TLR system, and actually it's all TLR that are being used. It's the MPL from the cell wall in the JSK system, and it's the TLR7 and the TLR9 agonists in the in some of the coronavirus vaccines. Let's now move a little bit on to the vaccination outcome. And of course, obviously, what you see here on the left hand on the y-axis is more or less the severity of disease as it, and here I have given a kind of a threshold. So if you want to prevent infection and disease, you best go for a vaccine that prevents infection. It looks very clearly that this is not the case, that you can achieve that currently with the current vaccines against coronavirus, because the pre-existing neutralizing antibodies are there. You can measure them, they can neutralize in vitro, but we have this phenomenon, I guess that has been discussed before, of the respiratory tract mucosa that is so large that you do not have sufficient amounts of antibodies there. Obviously, if you can prevent infection, you can also prevent transmission and disease. And you could argue, so that's the perfect vaccines. I wouldn't agree really, because no booster by natural infection is a major drawback by this type of vaccines. And therefore, you are really depending on boosters by vaccinations. And in many cases, this is considered a strong argument against or a con for prevention of infection. And obviously also there is a single spot or a few spots only that will therefore also allow for the emergence of resistant or variants that are resistant against the immune response. The next type would be actually prevention of stable infection, which is something that occurs with numerous vaccines, and that's something that is really preferred. And we see that also, at least in part, with the corona vaccines, that's based not only on neutralizing antibodies, but also some T-cell responses, and clearly also antibodies that work through antibody-dependent killer mechanisms, be they by complement neutrophils or ADCC-like. So you have partial protection against transmission, you have protection against disease, and you get a partial booster. And I think that is a good compromise for many vaccines. Ultimately, of course, vaccines should prevent disease or at least ameliorate disease. And that clearly depends not only on antibodies, but so on T cells, and there may be actually room for improvement through CTL responses, as Hans-Georg showed so nicely in his kind of vaccine. And there may also be a further improvement through unconventional T cells of the mucosa-associated immune system, and perhaps also trained immunity. Here you get a broad immunity based on the T cells, reduced breakthrough by variants, and unfortunately, obviously, infection and transmission occurs, and therefore new variants play a major role. Let me point something out, which is also kind of reinventing the wheel from vaccinology, and that is the use of correlates of protection and surrogates of protection. Now, a correlate protection in its strictest sense is defined as a correlate that really is responsible for protective immunity and therefore neutralizing antibodies for POI kind of fulfill that requirements, but it's also not fully fulfilled if you look at variants, as I will show in a minute. That's much more difficult for POD, prevention of disease correlates, and actually indeed we do not have this kind of correlates in most vaccines where T cells play a role, including the coronaviruses. So we go to surrogates of protection. That's more or less an immune response or a response in generally that is constantly seen in protected individuals, but absent in unprotected individuals. And that is best seen, obviously, in controlled studies, be they clinical trials or controlled observations. But again, quantity as above here also plays a role. And that would be antibody-mediated killer mechanisms, T cells, and most recently biosignatures, as is being done in cell vaccines now, where you look at ideally even single cell biosignatures. So with regard to this topic on correlates protection and correlates surrogates of protection, let's look briefly on the situation with the variants where we have nice in vitro assays, and those were discussed in different variants before. What we see here is clearly that after a third dose in this particular study, you still get full protection of infectivity in vitro. But in real life, we see after the third booster, that slide was been shown before, we see clearly that in real life, this does not translate in full protection against infection or even symptomatic disease. But fortunately, we see clearly that protection against hospitalization, that is severe disease, we still find these high proportions. So here we see a correlate, if you want so, but I would rather say a surrogate of protection in the neutralizing antibodies, because it's quite clear that the neutralizing antibodies will not suffice for prevention of hospitalization, but it would, like shown here in a very simplified way, they contribute obviously to blocking entry, but you would also want T cells and other antibodies to get this kind of prevention of severe disease as measured by hospitalization. And that already, I think, brings me to the end and looking forward, and I couldn't agree more to what Hans-Georg and others said. We really should look at the future in a sense that we should, of course, as long as necessary, continue with revaccinations, with boosters, because by changing epitopes, notably in the RNA vaccines, that's a easy undertaking. But as Peter said, we are a step further now in the flu field after decades, don't forget that after many decades, towards universal vaccines. And I think that's the way to go also for the pan-corona vaccines, where we would like to have neutralizing antibodies to the spike RBD, ideally to more conserved regions than those that are prone for mutation and therefore variant evolution. We need broadly reactive antibodies. We know a lot about that from the HIV field, which go for neutralization and antibody-mediated killer mechanisms, complement PMNs typically or ADCC. We want CD4, CD8 T cells to conserve antigens epitope. And we may consider, as I said before, MAID cells, mucosa-associated immune cells, and perhaps also trained immunity to be in front and to be proactive rather than reactive. And I think I will finish here to kind of make a little bit of time after this wonderful but rather long discussion round today. Thank you.
Thank you so much, Stefan, for this very insightful actually overview on all of the vaccination types and the aims we should have with vaccination. It's open for discussion, actually. So any remark? Let me have a look at the chat. No, there's not. I was just, maybe I start just with, did I get that right? That you said that one should also give the immune system enough time actually to optimize the immune response and develop broadly reactive immune responses rather than coming again and again with boosters and challenging the immune system with varied variants of concern, so to speak, again and again, and vaccinating behind the pandemic, so to speak. Is that correct?
Well,
so that boostering doesn't make much sense.
Well, I think everybody would agree on it. It's more the kind, and that is where the science comes in to see how we can actually speed up the process. I mean, if we go on the canonical way, and I didn't discuss that today, but I would really think that we should go for more adaptive trial designs. In that case, aside from the early preclinical studies, go into adaptive trial designs. And actually in the COVID field, it looks like our regulators are getting much more open in that discussion. I'm not looking at the pie at all. They were very, very helpful often in the last 2 years. But on general speaking, yes. I mean, what I didn't really spell out, I mean, the question also is, obviously, are we happy with clinical trials as they are now done in the Valneva case, where you just look for so-called correlates of protection? That I'm a little bit doubtful whether that is the full solution to it, but that's how it's going. And in principle, still, I would go for adaptive trial designs.